So chapter number 4, Animal Kingdom. When you look around, you will observe different animals with different structures and forms. As over 1,000,000 species of animals have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. Basis of classification. In spite of differences in structures and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, pattern of digestive, circulatory or reproductive systems. These features are used as the basis of animal classification and some of them are discussed here. So now, levels of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates, that is, they exhibit cellular level of organization. Some division of labor activity occurs among the cells. In coelentrates, the arrangement of cells is more complex. Here the cells performing the same function are arranged into tissues, hence is called the tissue level of organization. A still higher level of organization, that is the organ level, is exhibited by members of platyhelminths and other higher phyla, where tissues are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, mollusks, echinoderms and chordates, organs have associated to form functional systems, each system concerned with a specific psychological function. This pattern is called the organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. For example, the digestive system in platyhelminths has only a single opening to the outside of the body that serves as both mouth and anus and is hence called incomplete. A complete digestive system has two openings, mouth and anus. Similarly, the circulatory system may be of two types. First, open type, in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it. Or second number, closed type, in which the blood is circulated through a series of vessels of varying diameters, that is arteries, veins and capillaries. So after the levels of organization, now it's time for the symmetry. So figure 4.1 radial symmetry and figure 4.1 part B bilateral symmetry. It means divided into two halves and here divided into many halves. Now symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical, that is, any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of the body divides the organism into two identical halves, it is called the radial symmetry. Coelentrates, ctenophores and echinoderms have this kind of body plan. Animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. Now, diploblastic and triploblastic organization. So, animals in which the cells are arranged in two embryonic layers. Okay, an external ectoderm and an internal endoderm are called as diploblastic animals. Example, coelentrates. An undifferentiated layer, mesoglia, is present in between the ectoderm and the endoderm. In the figure 4.2, you can see that there is a ectoderm, endoderm, this is the ectoderm, endoderm, and in between there is a mesoglia. So these are diploblastic. Those animals in which the developing embryo has a third germinal layer, mesoderm, in between the ectoderm and endoderm, means in between ectoderm and endoderm there is third germinal layer, mesoderm, are called triploblastic animals, platyhelminths to chordates. In the diagram here, the ectoderm, endoderm and in between the mesoderm. These are triploblastic organisms. Now another factor, coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity, which is lined by mesoderm, is called the coelom, Animals possessing coelom are called coelomates. Example, annelids, mollusks, arthropods, echinoderms, hemichordates and chordates. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and the endoderm. Such a body cavity is called pseudocoelom, and the animals possessing them are called as pseudocoelomates. Example, aschelminthes. The animals in which the body cavity is absent are called as acoelomates, platyhelminths. So means coelomates, pseudocoelomates and acoelomates. Acoelomates means platyhelminths. So here in the figure 4.3, first are the coelomate, in which there is a proper coelom, meaning cavity, which is lined by the mesoderm. In pseudocoelom, there is mesoderm present, but in this form of scattered pouches. So called as pseudocoelom. Example is the aschelminthes. So obviously only one example. So next one is the acoelomates. Example, platyhelminths. There is no mesoderm. Next one is the segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomena is called as the metamerism. Now next, notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development. In some animals with notochord are called chordates, and those animals which do not form this structure are called as non-chordates. Example, porifera to echinoderms. Now classification of animals. The broad classification of Animalia based on the common fundamental features as mentioned in the preceding sections is given in figure 4.4. So here's the figure 4.4. Broad classification of kingdom Animalia based on the common fundamental features. So first we are starting with the kingdom Animalia on the basis of level of organization, cellular level and tissue organ or organ system level. Cellular level is given to the porifera and others, Cylindrita, Ctenophora, Platyhelminths, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, Chordata are in the tissue organ and organ system level. Now radial and bilateral symmetry. On the basis of this, radial, Cylindrita, Ctenophora and bilateral all others. Now coming to the acoelomates, pseudocoelomates and coelomates. Acoelomates is only platyhelminths, pseudocoelomates only aschelminthes, and others are on the higher level are like annelids, arthropoda, mollusca, echinodermata, hemichordata, chordata are the coelomates. So this is a basic classification, very basic level but important. And now it's time for the starting of different phyla. So the important characteristic features of the different phyla are described. So first is the phylum Porifera. Members of this phylum are commonly known as sponges. They are generally marine and mostly asymmetric animals. These are primitive multicellular animals and have cellular level of organization. Sponges have a water transport or canal system. Water enters through minute pores, ostia, in the body wall into a central cavity, spongocoel. So sponges have a water transport or canal system. Water enters through minute pores, ostia, in the body wall to a central cavity, spongocoel, from where it goes out through the osculum. So enters through the ostia and exit through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange and removal of waste. Choanocytes or collar cells, again choanocytes or collar cells line the spongocoel and the canals. Clear? Digestion is intracellular. The body is supported by a skeleton made up of spicules or spongian fibers. Sexes are not separate. These are hermaphrodites. That is, eggs and sperms are produced by same individual. So hermaphrodite. Sponges reproduce asexually by fragmentation and sexually by formation of gametes. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult. Now NCERT examples in the figure 4.5. Examples of porifera: Cycon, Euspongia and Spongilla. So Cycon, Euspongia, Spongilla. So ये चल के जा रहा है यहाँ पे Spongilla, यहाँ पे Euspongia, इससे बात भी करते नहा सकते हैं, इस तरह का कुछ लग रहा है ये। एंड दिस इज द साइकन। सो साइकन, यूस्पोंजिया एंड स्पॉन्जिला। सो साइकन, स्काइफा, स्पॉन्जिला फ्रेश वाटर स्पंज, यूस्पोंजिया बाथ स्पंज। तो नहाते हैं भाई, साइकन, स्काइफा, यूस्पोंजिया बाथ स्पंज, स्पॉन्जिला फ्रेश वाटर स्पंज। साइकन, स्काइफा, यूस्पोंजिया बाथ स्पंज एंड स्पॉन्जिला फ्रेश वाटर स्पंज। सो साइकन, यूस्पोंजिया एंड स्पॉन्जिला। नाउ फाइलम सिलेंट्रेटा यानी कि निडेरिया। दे आर एक्वाटिक, मोस्टली मरीन, sessile or free swimming, radially symmetrical animals. The name Nidaria is derived from the nidoblasts or nidocytes, which contain the stinging capsules or nematocyte present on the tentacles of the body. So body have nidoblasts or nidocytes in which the stinging capsule or nematocytes are present. So nidoblasts are used for anchorage, defense and for the capture of prey. Anchorage, defense and for the capture of prey. Cnidarians exhibit tissue level of organization and are diploblastic. They have a central gastrovascular cavity with a single opening called hypostome. So digestion is extracellular and intracellular both. Some of the cnidarians, example corals, have a skeleton composed of calcium carbonate. Cnidarians exhibit two basic body forms called polyp and medusa. The former is a sessile and cylindrical form like Hydra, Adamsia, etc., whereas the latter is the umbrella-shaped and free-swimming like Aurelia or jellyfish. Those cnidarians which exhibit in both form exhibit alternation of generation, metagenesis, that is polyps produce medusas asexually and the medusae produce polyps sexually. Example, Aurelia. So here is the polyp form and there is the medusa form. So this is the medusa form of Aurelia and this is the polyp form of Adamsia. Examples very important: Physalia, Portuguese man of war; Adamsia, sea anemone; Penatula, sea pen; Gorgonia, sea fan; Mandrina, brain collar. So Physalia, Portuguese man of war; Adamsia, sea anemone; Penatula, sea pen; Gorgonia, sea fan; Mandrina, brain collar. Once again, Physalia, Portuguese man of war; Adamsia, sea anemone; Penatula, sea pen; Gorgonia, sea fan; Mandrina, brain coral. And here is the diagrammatic view of a nidoblast. You can see the different organs. This is actually a cell. So now is the phylum Ctenophora. Ctenophores, commonly known as sea walnuts or comb jellies, are exclusively marine, radially symmetrical, diploblastic organisms with tissue level of organization. The body bears 8 external rows of ciliated comb plates which help in locomotion. Digestion is again both extracellular and intracellular. Bioluminescence, the property of living organisms to emit light, is well marked in ctenophores. Sexes are not separate. Reproduction takes place only by sexual means. Fertilization is external and with indirect development. Examples: Pleurobrachia and Ctenoplana. Pleurobrachia, Ctenoplana. So this is the example of Ctenophora means Pleurobrachia. So this is the Pleurobrachia. यहाँ पे आपको 8 complex easily दिख जाएंगी. So now it's time for the platyhelminth. They have dorsoventrally flattened body, hence called flatworms. Dorsoventrally flattened body. These are mostly endoparasites found in animals including human beings. Flatworms are bilaterally symmetrical, triploblastic, acoelomate animals with an organ level of organization. Hooks and suckers are present in the parasitic forms. Some of them absorb nutrients from the host directly through their body surface. Specialized cells called flame cells help in osmoregulation and excretion. Sexes are not separate. Fertilization is internal and development is through many larval stages. Some members like Planaria possesses high regeneration capacity. Examples are Taenia, tapeworm, Fasciola, liver fluke, and also Planaria is here. And the diagrams. This is the tapeworm, obviously, large length, and this is the liver fluke with hook or sucker. Now phylum Aschelminths. Aschelminths. The body of Aschelminths is circular in cross section, hence the name roundworm. They may be free living, aquatic and terrestrial, or parasitic in plants and animals. ठीक. Roundworms have organ system level of body organization. They are bilaterally symmetrical, triploblastic, pseudocoelomate animals. So alimentary canal is complete with a well developed muscular pharynx. An excretory tube removes the body waste from the body cavity through the excretory pore. Sexes are separate, dioecious, that is males and females are distinct. Difference भी होता है. Often females are longer than males. Fertilization is internal and development may be direct. The young ones resemble the adult, or indirect, जिसमें कि larva वगैरह बनता है. As examples are Ascaris roundworm, Wuchereria filarial worm, and Encyclostoma hookworm. So Ascaris roundworm, Wuchereria filaria worm, Encyclostoma hookworm. Ascaris roundworm, Wuchereria filaria worm, Encyclostoma hookworm. So this diagram figure 4.10 is basically given to show the distinction between the males and the female. That is because these are dioecious. So Ascal means roundworm, this is the male, obviously shorter, and female longer and slightly larger looking.